Hello, everybody. Welcome to the Closer Look Journal Club. I'm joined here today by my co-hosts, Manuel Rausch, Adrian Buganza-Tepoy, Matt Bursey, and Johannes Weikenmeyer. And today we're really excited to host Brianne Connizzo. Brianne completed a bachelor's degree in engineering at Smith College and then completed her PhD in bioengineering at the University of Pennsylvania. After that, she moved to Boston for a postdoc at MIT and then started as an assistant professor in biomedical engineering at Boston University in 2021. And in her lab, Brianne does really interesting work towards understanding soft tissue aging, where she pulls from multiple experimental techniques and, as I'm sure we'll see in this talk, does a really beautiful job of investigating the connection between biological structure and content and mechanical function. I'm really looking forward to learning more about her work. So Brianne, please go ahead.
Thanks so much. And thank you so much for the organizers for having me here. I'm really excited. I've been to a couple of these and it's always so great to see the research that's going on. So today I'm going to be talking about some work investigating the structure and function in different regions of the periodontal ligament, which I'll refer to as the PDL. And I want to start off just by saying that this is a true collaborative effort between me and Dr. Gili Naveh, who is a dentist at Tufts University, used to be at Harvard University. And so this is really a true collaborative effort. It was really Gili's idea to start this work, and she's an expert in the non-uniformity of the PDL, and I kind of came in on the back end doing a lot of the biomechanics work, given my, my research in tendon and ligament biomechanics. So just to start with a little background, the periodontal ligament is a fibrous structure that connects the tooth to the mandible. And you can see it denoted in this image on the right by this kind of pink striped structure here. The function of the PDL is to stabilize the tooth during chewing and grinding. And in particular, it acts as a shock absorber, preventing movement in response to high abrupt forces during chewing. The PDL also plays a central role in adaptation to load, facilitating longer-term bone and ligament remodeling during development, as well as during orthodontic tooth movement or when you have braces. This complex function is enabled by a three-dimensional structure, which is really unique and sets it apart from other ligaments. So shown here is a 3D volume rendering micro CT with contrast enhanced of a sagittal section on the left and a transverse view on the right, where you can see the bony mandible as well as the structure of the tooth with the pulp and the root. And surrounding the tooth, you see highlighted in green, the periodontal ligament. And I want to point out here that, you know, unlike other ligaments, it's not just a direct connection from one attachment to another attachment. It's really a three-dimensional volume, kind of like a capsular structure that directly and completely envelops the tooth. And you can see on the right this characteristic figure 8 shape that the periodontal ligament forms around the two roots, and this is a molar. So we're really interested in two different regions of the periodontal ligament that have significance for biomechanical function. The first region is called the dense collar, and this region was identified and really beautifully characterized by Gili back in 2018. The dense collar is the topmost or the most coronal 200 microns of the periodontal ligament, and it's characterized by really dense organized collagen fibers, as you can see with the picoserous red staining on the top right here, and then on the bottom right, which is a collagen fiber density map where the densest regions are shown by the yellow regions. And it also has kind of a traditionally ligamentous structure, so a single bony attachment on either side and dense organized fibers that span between. And you can see the preferential fiber direction in the picoserous red staining. Another region that's of interest to us is just underneath the middle of the two roots, and this region is called the furcation. And the furcation is interesting because unlike the collar, it actually has sparsely disorganized collagen, as you can see in the maps on the right. And there's also other tissue structures here, such as blood vessels and potentially nerves. And so it's kind of a complex environment that is exhibiting different mechanical properties, as I'll show you later in the talk, but also potentially an area for lots of remodeling and lots of nutrient transfer. So, But beyond this, beyond this really nice characterization of collagen fiber structure, we really don't know very much about ECM composition, specifically, what other types of ECM components are there, and particularly how it relates to mechanical function. So the first part of this talk is really the focus of the paper that I was selected for this journal club, which is trying to understand the regional composition in these particular regions and then relate it back to mechanical function. So to do this, we harvested mandibles from 9-week-old male mice. And then we took regional specimens using laser microdissection from the collar region, the furcation, and then we also took regions from the apex, which is just at the very bottom of one of the roots. These small pieces were then processed for liquid chromatography mass spectrometry, and they were analyzed with Max BioCrown software with false discovery rates less than 1%. And then we also followed up this mass spec analysis with immunohistochemistry on whole embedded tooth specimens to both verify our results, but also further characterize these spatial patterns. So starting with this mass spec data, as you can imagine, there's lots and lots of proteins that are expressed throughout the periodontal ligament. And so we kind of characterized our analysis into comparisons between the collar and the furcation, the collar and the apex, and the furcation in the apex. And just to orient you to how the data will look, the higher abundance in the first of the two paired comparison will be shown in green, where the bar represents the size of the fold change between the collar and the furcation in this case. And then the yellow bars on the bottom will represent the higher abundance in the opposite region. So everything on the top in green for this particular comparison is higher in the collar, and on the bottom in yellow is higher in the furcation. And we do this for our two other comparisons as well. So, in the interest of time, and I don't have time to talk about every single protein that we found, I'm just going to focus primarily on the proteins that are really relevant to biomechanical function, and those that were quite interesting to us. So the first thing we noticed was when we looked at both comparisons between the collar and the furcation and the collar and the apex, you can see that collagen 6 popped out as a huge difference between these two regions with high abundance in the collar. And you'll also find that other minor collagens such as collagen 5 and collagen 12 were also present. So collagen 6 is a really interesting one. It's a microfibril forming collagen, and you can see up in the top left here this really dark brown staining. So just to orient you, all of the immunohistochemical images will be shown with a purple hematoxylin counterstain, and then your brown is your DAB immunostain for the particular target that's shown up here in the top left. And then the T's represent the tooth, the B, the bone or the mandible, and the P represents the periodontal ligament. And so collagen 6 was present in high abundance in the collar region, and it's a microfibril forming collagen, which is responsible for increasing organization of type 1 collagen fibers. So not all that surprising, since this region of the periodontal ligament is actually quite organized and has densely organized collagen. It's also associated with anchoring the basement membrane of blood vessels, nerves, muscles to the ECM. So it could also play a role potentially in these other structures that might be present at and near the periodontal ligament. And I'll also point out that collagen 6 is a marker for the pericellular matrix. And so in other tissues like cartilage and tendon, we often see collagen 6 highly expressed, particularly around cells that exhibit high mechanosensitivity. Collagen 5 is also an interesting target to find in high abundance, and it was found mostly in the collar in our mass spec, but showed very uniform distribution in our immunohistochemistry. Collagen 5 is a minor collagen that's associated with fibrillogenesis. It's been studied very extensively in tendons because mutation in collagen 5 is actually associated with hyperelasticity, and it's the primary cause of Ehlers-Danlos syndrome, which is a hyperelastic connective tissue disorder. Collagen 5 in particular regulates fibril size and distribution. In particular, if you do not have collagen 5, you get larger fibrils, and that can change, obviously, the mechanical function of the tissue. So in addition to these minor collagens, another interesting target we found that was higher in the furcation was TGM3. And that was actually present in all three of our tissues. So it was present in the collar, the furcation, and the apex, but it was highest in the furcation compared to both other regions. TGM3 is part of the transglutamase family. And these are enzymes that speed up chemical reactions to break down tissue. So one of the most famous one is amylase, which is often found in your saliva to break down tissue, right, for when you're eating. And this could potentially indicate a really high remodeling rate due to damage that's happening in that area. So we know that this area of the periodontal ligament is experiencing high compressive loads because obviously as you chew, the tooth is being pushed down and the periodontal ligament is compressed between the tooth and the bone. And so this might indicate that there's high remodeling here in the periodontal ligament. But interestingly enough, we also found that there were proteins associated with bone remodeling in this region that are listed there. And so there's some evidence that perhaps the periodontal ligament and the underlying alveolar bone there are interacting and kind of communicating with each other to remodel effectively to withstand that high compressive environment. We also found lysyl oxidase abundant in the periodontal ligament kind of throughout, but especially in the furcation. It's a little hard to see here because it's so small, but it's actually quite dark in that region. And lysyl oxidase is really intimately involved in the production of crosslinks for extracellular matrix proteins. So it's really heavily involved in collagen cross-linking. And actually other studies have shown that tendons and ligaments don't really develop properly without lysyl oxidase. And in fact, their mechanical properties are extremely weak. One interesting finding that we had was that we actually saw that there were these longitudinal structures in the periodontal ligament, particularly in the dense collar. And these structures stained really strongly for lysyl oxidase. And we think that these, the longitudinal structures might suggest some sort of anchorage or reinforcement, particularly in that region, because it's an area of high tensile force, but also torque and shear. There were a bunch of other findings that I would love to discuss if we have time after, but in the interest of time, I really just focused on the ones that were relevant to ECM molecules. So just to summarize kind of the mass spec data that we have, before we did the study, we obviously knew a lot about the collagen organization, showing that there were these dense collagen fibers in the collar and sparsely present fibers in the furcation. And we knew a little bit about blood vessels present, but we gained a lot of insight in this compositional study to other proteins that are present here. So particularly in the dense collar, we found the expression of minor collagens supporting fibrillogenesis, as well as some other proteins suggesting it may be a region of reduced cell density and cell migration, which kind of supports its barrier function. And then in the furcation, we found an increased expression of cross-linking enzymes, increased bone remodeling, and expression of mineralization markers. And so this suggests kind of a hybrid tissue where you have expression of not only ligamentous genes, but proteins, excuse me, but also communication with the underlying bone. But the overall goal of doing this compositional study was actually to figure out how this actually relates to the mechanical function of PDL. Right. And so in order to do this, I think what we wanted to do was actually be able to measure the mechanical function of PDL in different regions. Right. And this is actually quite complicated because the mouse PDL is really small. Right. So it's only 70 microns across. It's really, really tiny. And you can imagine just how tiny a mouse tooth is. And so that's kind of where I came into the project with my expertise in AFM nanomechanics. Trying to adapt some of the work that we have done in other tendons and ligaments to performing it in the periodontal ligament. So what do we know about mechanical function of the PDL to begin with? So there are a couple, actually plenty of studies that have investigated tensile mechanical properties. So in tension, we know that the PDL exhibits characteristically ligamentous mechanical behavior. And it really exhibits a nonlinear stress strain curve with rate dependent behavior, very similar to other ligaments like the ACL or the ulnar collateral ligament. Mechanical behavior of the PDL has really been tested quite a bit in multiple teeth, multiple root levels, and multiple animal models. But much of the work has really been done in large animal models, and a lot of it has been done in such a way where you have to actually remove the periodontal ligament from the tooth itself, right? So in these large animal models, you basically cut a piece of the bone and cut a piece of the tooth, and then you can put it into your traditional instron tester and measure tensile mechanical properties. And so across multiple teeth, multiple root levels, and multiple animal models, there was a wide range of elastic moduli in the literature. And this supports really heterogeneous properties. Not only are you testing different parts of the PDL, but also across these different variables. One thing that was kind of missing is that dynamic mechanical behavior wasn't really done experimentally. It was really often characterized using constitutive and finite element models. So we learned a lot from these finite element models. However, they often address the PDL as a uniform tissue, which computationally obviously makes things a lot easier. But from our studies with structural analysis and compositional analysis, we know that the PDL really isn't a uniform tissue. Regional differences along the collar. So even if you're looking at just the figure 8 structure of that collar within a root level, really those regional differences had never been explored before. And the mechanical function of the furcation was also not really well understood. So despite this extensive literature on PDL mechanics, there were really very few studies that have investigated the regional differences in tissue function, and none of them were able to do this in situ. And so that was the primary goal for our work, to figure out a way to test regional properties without disrupting the native structure of the PDL. So I came into this project because I had a background in using a nanoscale rheology technique to understand tendon poroelasticity. So I just want to talk a little bit about the background of this technique and what the advantage is of it to understanding nanomechanics. So essentially, we have a AFM-based rheology technique where we can measure viscoelastic and poroelastic properties independently. And the way that we do that is that in the commercial AFM head, we can apply a pre-indentation that's at the micron scale using the piezoelectric device that's naturally in the system. And then we can allow our tissue to relax. And then we apply with a secondary piezo that's located underneath the sample with a custom setup, we can apply a nanoscale dynamic frequency sweep. So for those of you who are kind of familiar with this technique, essentially you input, a dynamic stress, a sinusoidal stress, you get out a sinusoidal strain, and then the ratio of the amplitudes of the stress and strain is equivalent to the dynamic modulus of the tissue. And then the phase lag between the two is indicative of the phase angle. And the dynamic modulus can be equated to, the storage and mechanical properties of the ECM itself, while the phase angle is often attributed to viscoelastic or fluid flow. And so it turns out when you use this technique, what you're able to get is this full frequency spectrum response, where you can measure the modulus and the phase angle at any frequency from about 1 Hertz to 10 to the 3 Hertz. And so at every single one of these frequencies, we're able to extrapolate this really nice curve. And when we did this with cartilage, actually this work was done by Hadi Nia back in 2011, well before I was in the lab. He found this really beautiful single peak in the phase response, which you can see right here. And it turns out that this single peak is actually movable. So if you increase the depth at which you're indenting, or you increase the size of the particle that you're indenting with, you can actually shift this peak to the left or the right. And this shift has a linear relationship where the frequency of that peak is directly and inversely related to the contact area, so the area of which you're contacting with. And so that actually, it turns out that follows classic poroelastic behavior. So if you remember back from hydrology or from permeability research, what this means is that it's directly related to fluid flow throughout the tissue. Interestingly enough, when we do this in tendon, we actually find this really interesting 2-peak phase response, where we have a linear poroelastic peak at this really high frequency around 10 to the 3. And you can see that it demonstrates this linear behavior as the cartilage peak did. But then we also have this non-poroelastic, purely viscoelastic peak that happens around 10 Hertz. And so this is nice because it allows us to independently measure the poroelastic peak, which is directly relevant to fluid flow through the tissue, and the viscoelastic peak, which we equate to native viscoelasticity of the fibrils themselves, as well as collagen reorganization such as fibril uncrimping, fibril sliding, and then fibril realignment. So using this technique, we had published a couple papers showing that with aging, poroelasticity in tendons is altered, and also with different regions of the tendon. And so Gili came to our lab asking if we could potentially use this to understand poroelastic and viscoelastic behavior of the periodontal ligament. And so that's what we did. So we adapted this technique to use in an in situ PDL sample. And so I'm going to talk you through the procedure. And I want to point out here that the procedure to actually do the indentations took a lot longer than the data processing, and I'll tell you why in just a second. So to embed these samples, we took hemimandibles and embedded kind of a multi-tooth section in cryosectioning media. We then cryosectioned from the apical surface until we were right at the level where you could be have both roots and also see the PDL with both furcation and the collar present. We flipped the sample over, bonded it to an AFM stage using dental composite, and then we were able to indent the PDL at different regions over the whole length of the tissue without disrupting the outside bone or the inside tooth. And this technique actually took a very long time because, as I mentioned before, the periodontal ligament is quite small, it's only 70 microns, and the visualization is quite challenging. So this image was taken through a microscope, and you can see that we have stained the two roots of the molar with Verhoff stain, and that allowed us to be able to identify where the center of the tooth is so that we could essentially develop a protocol so that we could indent the periodontal ligament in multiple directions. And so this is the protocol we came up with, and this was by necessity. We ended up indenting 29 different regions because, quite frankly, we weren't sure where the ligament was going to be, right? So because it's so small, and the AFM tip that we use for this technique is 25 microns, there was a good chance that you, if you just tried to do it by eye, you might not hit the actual ligament. And so the way we got around that was by essentially starting at one of the roots and traveling down and indenting every 50 microns from a point at which we were in the pulp of the tooth to a point at which we were definitely in the bone of the mandible. And we did that in the distal lingual, so lingual being tongue, buccal being cheek, mesial towards the head, and distal towards the mouth. So we did that in the distal lingual region, and then we indented on the other side in the distal buccal region. We came back to the root and went across the center to indent the furcation, and then again on the mesial side to do the mesial lingual region and the mesial buccal region. And so what we ended up with is 29 different regions, and for each of those regions we had at least 3 indentations that we averaged, and for each of those indentations we had curves that looked like this. And so from these curves, we identified the viscoelastic and poroelastic peaks. We measured the low frequency modulus, which is similar to an indentation modulus, so static modulus. We also measured the high frequency modulus, which was defined as the modulus just after the poroelastic peak. And then we used those 2 moduli values to measure the self-stiffening ratio. And so tissue that self-stiffens basically means that the faster that you indent it, the stiffer it seems, or the stiffer the mechanical properties are. And then we also measured the frequency and phase angle of the viscoelastic peak, as well as the frequency and phase angle of the poroelastic peak. So a total of 7 parameters that we measured for each region. And then we average indentations within the position and then pooled within regions. I'll talk about that in a second. And then made statistical comparisons between them with t-tests. So the first step that we had to take in this was to identify the tissue that we were indenting, right? So as I mentioned, we took 29 different regions, and within each of those 5 areas, we had to identify what was ligament-like or periodontal ligament and what was not. And so one way that we did this was we made criteria based on indentations that we had performed using this system in the past to identify areas that were cartilage-like, ligament-like, and bone-like. And so anything that had a single peak in the phase response and an increase in self-stiffening that was above a certain threshold would be identified as cartilage-like. Anything that had a high stiffness as well as a low poroelastic behavior or a low phase angle behavior would be characterized as a bone-like material. And then anything that had a 2-peak phase response with a moderate self-stiffening ratio was identified as a ligament-like region. And so for each of our 29 points, you can see that we measured for all of the indentations that we made, you can see that we measured the number of bone-like, cartilage-like, and ligament-like regions in each of those 29 points. And then we identified regions that were mostly ligament-like. And so anything where there was greater than 50% of the regions were the greens. The green is ligament-like, the yellow is cartilage, and the red is bone-like. Anything where there was 50% green, we identified that as a periodontal ligament region, and that's represented by these stars over here. So for the rest of the presentation, when I'm referring to data from the distal lingual region, for example, I'm taking these 3 points, which happen to be indentations 4, 5, and 6. Okay, so the first thing we did was we compared properties between the collar and the furcation. So on the left in blue is the furcation, and on the right is the collar, and this is pooled over all of the different regions that we measured. And shown on the left is the low frequency modulus, and on the right is the high frequency modulus. And so as you can see, in the collar compared to the furcation, there was a significantly higher modulus value for both the low frequency and high frequency moduli, suggesting that the collar has higher mechanical properties than the furcation, particularly in our indentation, which is indicative of compression, but also tension due to the way that we're indenting. But that doesn't tell the whole story, right? And so because we were able to capture this data from the mesiolingual, distal buccal, and mesiobuccal, we then wanted to see if there were any differences just within the collar that could explain that wide range of variability that we found in the collar. And so I'm showing you the same exact data here. The low frequency modulus is going to be on the left and the high frequency on the right. And now we're showing the furcation again in blue, and then the mesial lingual and distal lingual, so the lingual portions are in red, and then the buccal portions are in yellow. And I think what you can appreciate from both of these plots already is that there is significant heterogeneity even within the collar. And so you can see that most of the difference between the furcation and the collar is actually due to the buccal regions. The lingual regions were actually quite similar in terms of moduli values, both low and high frequency, and the buccal regions were stiffer. And interestingly enough, we also found that there was lingual buccal differences, right? So both at the mesial and the distal regions, the buccal was significantly higher in modulus than the lingual. And so this told us that much of the collar furcation differences, in terms of modulus at least, are due to the buccal side of the collar and not the lingual. When we looked at self-stiffening ratio based on region, which I'll remind you is the ratio of the high frequency modulus to the low frequency modulus, we also found some interesting things. So the collar was lower in self-stiffening ratio than the furcation, but you can see this wide variability in the furcation. And the mesiobuccal and the distal lingual were actually the lowest, whereas the outside regions, the distal buccal and the mesiolingual were higher. And that was really interesting because it suggests that there is actually significant buccal lingual non-uniformity in self-stiffening. So differences between the lingual buccal at the mesial side and also the distal side. When we look at the viscoelastic peak data, which I'll remind you again is indicative of matrix viscoelasticity as well as reorganizations, we did find that the mesial collar and the furcation were significantly higher in phase angle than the distal. but we didn't find anything in frequency. So overall, it suggests that viscoelasticity varies with regions and is generally higher on the mesial side, as well as the furcation. And I know I'm going through a lot of data at this point. I will provide a summary in just a little bit, I promise. When we looked at the poroelastic peak, which again is indicative of fluid flow and permeability, we found that the mesiolingual and the furcation sides were highest in both phase angle as well as in peak frequency. And we also saw a peak frequency increase in the distal lingual compared to both of the buccal regions. And so this suggests that permeability or poroelasticity is highest on the lingual side as well as in the furcation. So just to summarize kind of what it all means, I've created this, this summary graph where what you can see is the self-stiffening ratio shown in yellow or brown, where the high values are shown in the darker color and the low values are shown in the lighter color. The modulus is then shown in blue, and then the permeability is shown in purple or pink. And so compressive modulus we found was higher in the collar than the furcation. And so we think this is likely due to the increased density of the collagen fibers there, and also the strength and tension that's required to prevent tooth from moving too far down during chewing. It's not super surprising, but it is nice to see that our mechanical property measurements do match what we're finding in, structure and composition. Interestingly, we found that the compressive modulus was higher on the buccal side than the lingual side. And this has really not been found by any other mechanical study before, but it actually makes sense. So there is a slight tilt of your teeth towards the buccal surface. And so this naturally creates more compressive loading on the buccal side. And so it does actually make sense that the modulus would be higher on the buccal side. And it was nice to see that our mechanical evaluation, which was totally unbiased, actually represented the loading environment that you would expect in the mouth. Our self-stiffening properties were highest in the distal buccal over here, and the mesiolingual regions. And this is interesting because you see that it has kind of like an X pattern. And this suggests rotational self-stiffening about the collar, right? So if we see higher self-stiffening ratios in these two regions compared to these two regions, what you would expect is actually that this is perhaps an adaptation to rotational loads, or maybe torque that's happening present in the molars, of course, because we do a lot of grinding in the molars. It could be reflective of this higher frequency chewing and grinding that relies and requires this resistance to torsion, just as I said. We also found self-stiffening was higher in the furcation region, which it wasn't totally expected. But we think that this might be due to the high frequency and magnitude of compressive forces in this region. So if you imagine the sagittal view of the tooth is being pushed down. And so in between the tooth and the bone, there's actually compression of the periodontal ligament. And so this kind of reflects a more cartilaginous-like tissue, which typically has a higher self-stiffening ratio. We also found hydraulic permeability values for all of our PDL regions were lower than tendon, what we found tendon in previously, but higher than cartilage. And so that's interesting because we haven't tested more traditional ligaments like the ACL or other ligaments, right, rather than tendons. It could suggest that the periodontal ligament is actually neither a tendon or neither a ligament nor cartilage. It's kind of a transitional tissue. We also found that permeability is higher on the lingual side and in the furcation, which is interesting because this could be indicative of more nutrient transport. So the lingual side being, where your tongue is. Obviously, there's also bacteria and spit that is on that side. And so it's potential that you require permeability on the lingual side because that's where all of the nutrients are coming in. It could also be a result of different other tissues in the furcation. So one thing I want to point out here is that, we know that there are other tissues such as blood vessels present in the furcation, and we haven't done any characterization of vasculature using this rheology setup. So we really don't know if the mechanical properties could be relevant to vasculature. So that's kind of a source of future study. So just to summarize a little bit, The furcation and color, I hope I've convinced you, are really dramatically different in composition, structure, mechanical function, pretty much everything that you can dream of. And the PDL, especially in the furcation region, we found interacts directly with the underlying bone and is really involved in bone remodeling. This was something that other studies hadn't really found in the past. And so it was really exciting for us to see that because we're really interested in understanding the mechanobiology of the PDL and how it actually remodels in real time. This data suggests that there is a key for PDL remodeling and facilitating potentially tooth movement, so in orthodontics and in development. And so that's kind of where we're going in the next set of studies. And also, I hope that I've excited you by the fact that there's really significant heterogeneity even within the dense collar. And so this really hasn't been studied at all before. And this is really consistent with both the orientation of the tooth and the expected forces that the PDL would be under in normal everyday chewing and grinding motions. And so one caveat to this, of course, is that this is likely to vary with tooth, right? So a canine and a molar and incisor, these are all likely going to have different properties, and particularly the rotational self-stiffening that we found here, that may be specific to molars and not present in other tissues. So it's probably worth exploring other teeth as well. So, talking about the next steps, what we've learned from this and where we're going from here, obviously we know that the PDL is a highly complex 3D tissue, and it seems from our studies that it really can't be classified by traditional ligamentous or traditional cartilaginous properties. So I like to say, it's a soft tissue kind of in its own ballgame. Understanding this aspect, understanding this heterogeneity is really essential to making new discoveries about tooth biology. And we're hoping that kind of more researchers come into this field and start working in this realm to really better understand the differences in PDL structure and how that relates to function. I think future studies should focus on characterizing differences, specifically, like I mentioned before, differences on teeth and root levels. So you'll see that in this study, we really focused on one specific root level where we could get the furcation and the collar, that traditional figure eight structure in the same area, but we didn't do any mechanical evaluation of the apex, for example, or kind of the regions along the PDL from the coronal surface down to the apical surface. So I think that would be kind of interesting and really important as we're talking about this as a three-dimensional capsular structure and not just one-to-one ligament. I think kind of following up some of our mechanical data by looking at specific compositional differences within the collar. So I think it'd be really great to be able to relate some of the mesial distal and some of the lingual buccal differences that we're finding with differences in composition, whether that's altered proteoglycan content or minor collagens or cross-linking. So hopefully that's something that we'll follow up on in the future. And then also trying to understand better the biological function of the different regions. So one thing I didn't really discuss at all is the periodontal ligament also serves as kind of has a role in bacterial, the prevention of bacteria into the tooth structure, right? And so in some ways, maybe the mechanical properties kind of reflect that, and having this kind of dense collar region that can prevent migration of cells and migration of bacteria potentially. But we haven't really explored that in the different regions of the collar and as well as along the surface down to the apex. Moving forward, we're also really interested in focusing on mechanobiology, so specifically how extracellular matrix remodeling occurs in the PDL. So we want to identify how this remodeling occurs and how it's related to remodeling the bone. So in particular, Gili's just been funded a large NIH grant to try and understand the process of tooth movement. And so with the hypothesis that that changes in the PDL could precede changes in the bone, the underlying alveolar bone, thus making the PDL potentially a source for understanding, potential diseases like gingivitis, as well as a potential site for intervention, right? So if we can understand how normal function, how the normal function works, we can also understand how disease happens, and then eventually this could potentially lead to therapies or drugs that we could test out. So that's pretty much all I have for you today. I'm happy to take any questions. I'd like to just really quickly acknowledge some of the organizations involved. So when this project started, Gili was working at Harvard in the School of Dental Medicine, and I was over in my postdoc at MIT. We have both since moved, but the collaboration has stayed. And so now Gili's over at Tufts and I'm at BU. I'd also like to acknowledge our funding sources. So Gili, this project and our future projects are both funded by the NIDCR, and my lab has been heavily funded by the National Institute on Aging as well. And so continuous support from the NIH has been really critical in getting this work off the ground. And then, just a shameless plug for my lab. We're, as Emma mentioned, we just opened up last January, so we've only been open for a year, but we're growing really quickly. And if you're interested more in mechanobiology, ECM remodeling, aging, and sex differences, I invite you to please check us out on Twitter at Connizzo Lab or at our website, connizzolab.com. So thank you to the organizers for having me, and I'm happy to take any questions you have. I'll stop sharing.
Yeah, that was fantastic. Thank you so much. I really appreciated the slides explaining your AFM method too. That was super helpful and just like very clearly explained. So thank you.
Thank you.
Yeah. Spencer, if you want to go ahead with your question, please.
Yeah, hi Bree, great work. And I think I maybe just finally learned how you say your last name. I thought it was Connizzo and you said Connizzo. Is that right?
Yeah, it's Connizzo.
Okay, well, it's good to learn that.
It's a common mistake, don't worry.
Okay, this was fascinating. I had two questions I want to ask you. So one was, I know people talk about differences with mice compared to humans in all kinds of different studies, but I was curious about that with kind of impact of them eating. I think Mariana had a question about this in the chat too, but like eating the same thing versus differences in all the kinds of things we eat, if that impacts kind of forces on the teeth and how that would change things. Like, I guess I'm just curious in this context, what you think about mice compared to humans. That's my first question.
Yeah, I mean, that's a really interesting question. I think in terms of the forces that are being applied to the teeth in the molars, it's quite similar because they are, they're often doing the same types of motions, chewing, grinding, and the structure of the molars is quite similar. Other teeth, like for example, the enormous incisors that the mice have compared to humans. Maybe my co-author Gili can speak to that because she's the dentist in this respect. But in terms of the forces in the molars, it's actually quite similar. The other question you had though about the what they're eating themselves, that's really interesting too from like a nutritional perspective. Yeah, I don't really know much about that, to be honest. But it's a good question. I think.
Okay, let me ask another question related to that real quick, which is the forces that this ligament sees. I mean, is it mostly? It's not really tension mostly, right? Wouldn't it be a lot of shear and compression? I mean, do you have a sense of that?
Yeah. So from what we've done, it basically looks like the way that I envision it, at least, is it's kind of like a moon shoe. So you have like the tooth suspended in the periodontal ligament. So like the collar kind of acts like the rubber bands of the moon shoe where you push down, and the collar actually exhibits quite a bit of tension. And I think the furcation actually experiences quite a bit of compression. But they all kind of experience torsion and shear as it's a multi-axial with grinding, and obviously it's not like taking something into an intron and doing tension that way. So it's quite complex, but there is kind of a primary tensile area, which the collar seems to be it. And most mechanical studies of the periodontal ligament do actually test it in tension in that direction.
Very cool. Thanks. Great work.
Thank you.
Mariana, do you want to go ahead?
Sure. I really want to echo what Emma said. The slides were really beautifully put together and made it so interesting and easy to understand. Thank you so much.
Thank you. Appreciate it.
Yeah, I kind of wanted to go back to the question about loading, like Spence had. I put it in the chat. You know, like when I think of them like nibbling super fast, right? They kind of just get right at it before somebody eats them and then get out of there. And I was curious if, like, have people compared these ligaments in slow chewers versus fast chewers? Is there sort of any effort in the comparative biomechanics world to look at that?
You know,
That you know of, or even with, yeah, I guess that's
a really interesting question, especially in the context of what we're doing, because we're obviously doing like full frequency spectrum, right? And so we're looking at properties over different frequencies. So we often like think about what those frequencies really mean physically and how they relate to like what's going on. I don't think anyone has looked at that. I don't think anyone has. Well, I don't want to speak out of turn, but I'm not sure if anyone's quantified like chewing speed for different species and then related it back to PDL mechanics. As far as I know, at least in the biomechanics area of PDL, most of it has been like traditional tensile testing and not really dynamic testing at all. A lot of the dynamic work has really been done using models.
Right.
But yeah. It's a great question. I think it would be great to look at. I'd love to look at this in other animals and humans. It does seem that our data reflects very nicely the same types of forces that are being that you would expect from a human. You know, because like intuitively we know that this is what you're chewing and you're grinding, and those are high frequency forces. Maybe not high as frequency as what a mouse would be chewing at, but but it's a corollary.
Yeah, I mean, I love the idea of, trying to explain the heterogeneity that you see, right, in these tests and tying it back to some sort of functional level. And perhaps by broadening out the x-axis, you know what I mean, that we might be able to start identifying these trends, looking a bit more comparatively, right? So anyway, I think it's really fascinating. Great job. Thank you.
Thank you. Appreciate it.
Are there other questions from the audience? Yeah, Matt, please.
Yeah, sure. So first I want to say that I think this is the first talk, kind of like what Manuel said, it's the first talk that I've ever seen about teeth, especially in the biomechanics field. So I think it's like just really cool to, use a lot of the same terminology that we maybe think about on a daily basis, but apply to a completely different tissue. So I just want to say that first. But I had a question about the mass spec data that you presented at the beginning. And in particular, you talked about these sort of accessory collagens, collagen 5 and lysyl oxidase. And you mentioned like those are associated with connective tissue disorders like Ehlers-Danlos and others. So I was just curious, I mean, do people with those connective tissue disorders also have a tooth phenotype? Like are they more likely to lose a tooth than someone who doesn't have that mutation? Just out of curiosity.
Yeah, that's an interesting question. I actually did my dissertation on collagen 5 mutation, Ehlers-Danlos. I don't remember ever seeing anything about teeth, but it could be because that's such a minor thing that you wouldn't even report. I'm not really sure if there have been studies in that realm. Usually, with those types of connective disorders, there's larger scale problems, right? So oftentimes cardiovascular issues, skin lesions, stuff like that. And so those may overshadow, but I don't know if there's a dental phenotype in those. I don't know, maybe Gili have you ever heard of a dental phenotype in that?
Yeah, there are some dental phenotypes, especially some of them are more prone to gum disease, periodontitis, etc. I'm not sure if they looked at mobility or efficiency of orthodontic treatment, for example. These will be very minor, even if so. But there is probably some sort of a connection. But as Bree said, there are bigger problems. So the teeth are. And also in some of those syndromes, you also have problems in the eruption of teeth and the number of teeth. So the mobility is kind of the least problematic thing.
Sure. Yeah, I guess I'm just thinking about like mutations and how it may affect the PDL. And like, for example, like a lot of these mutations, there are animal models that, have a lysyl oxidase knockout or something like that. So there may be ways to use your technique in mice to potentially look at some of that. So just curious.
Yeah, I think it would be great. There are, there are available models that I have even used in the past that we could look at things like that. It would be pretty straightforward. But a lot of these models are embryonically lethal, or there are other issues. So you really need those targeted models, which exist for some. So like collagen has them. A lot of the minor collagens do have them, but not every ECM protein is going to have something like that. I'd love to look at those though. That could be something we talk about offline, Gili. Thank you though. Thank you.
Yeah, regarding the food, I have to just add, if I'm
Yeah,
speaking,
please.
they do eat hard pellets. And there is research that we looked at different diets and the structure of the periodontal ligament, and there's also a model which removed forces or loads from the teeth, from the opposing teeth. So they extracted the tooth, and then we're looking at the structure of the PDL. What happens is that it's I'm not sure if they looked at the stiffness itself, but the composition and staining. But what they saw is that, first of all, the periodontal ligament is becoming larger in volume. And then I believe it's less organized. So once you have loads, then the organization becomes much more organized. And when the loads are removed, the organizational level is much lower. That's what I remember.
Thanks, Gili.
Yeah, thank you. Yeah, one kind of random question, but how much are these ligaments straining under sort of physiological loading? I guess I'm actually just curious in humans as well, because like everyone's been saying, I feel like I haven't thought that much about teeth from a biomechanics perspective, but clearly it's a super rich problem.
Yeah, I mean, we're not talking about the strain that you'd have on an ACL, right? We're talking pretty small strains, and it's a pretty small ligament. There have been models to quantify like strain maps within a 3-dimensional. I don't remember what the exact numbers off the top of my head, but we're talking, similar to like other ligaments that are kind of within the toe region. I would say like 3-5% strain is what we're talking about on a regular basis with some frequency.
Got it. That makes sense. Still kind of like freaky to think about our teeth moving back and forwards. I guess you think about that a lot though on your end.
Yeah. And you really don't often think about like your teeth being movable structures until you're a dentist, right? Or until you need them to move, right? I mean, many of us have had braces, right? Which is literally you're moving your own teeth. So there has to be the functional mechanism to do so. But most people are not often thinking about how that happens and what the biology and the mechanics behind that are.
Yeah. Manuel, did you have a question or?
Very academic. I just wanted to highlight that dreaming about losing your teeth is a thing that I regularly do. And therapists will tell you it has to do with big life changes. So I'm wondering, after you started doing research on teeth, have those dreams occurred to you more frequently, and has it correlated with big life changes?
Well, I've had a lot of big life changes in the last couple of years since I just started my lab, and I actually had a child last year too. So I don't know if I can correlate the dental research with the teeth falling out as much as other things, but I'll keep you posted if it continues to happen. I'm gonna write them down now.
I have to mention that I saw that it's the most frequently dreamt, like most frequently horror dream that people dream all over the world, by the way. So it's not only you.
Yeah, my specialty is that I dream that they become soft. But anyways, neither here nor there. It's actually pretty common, so I'm not alone.
Yeah, it's very common.
All right. Well, maybe on that note, I'll thank our speaker for joining us today. Yeah, thank you so much, Brianne, and thank you, Gili, also for joining us. I think we all learned a lot from this talk, and we really appreciate you stopping by. And I can't remember off the top of my head who our next speaker is, but we'll be back in 2 weeks. So hopefully see you all there.
Really fantastic. Thank you so much.
Thanks, everyone.